Welcome to the BioInsights, where science comes alive.
Proteins begin as linear chains of amino acids, but folding transforms these chains into precise three-dimensional molecules. Understanding how proteins achieve their three-dimensional form is central to molecular biology and biochemistry. Proteins are constructed from 20 standard amino acids, each sharing a common molecular backbone. Their unique side chains, called R groups, distinguish one amino acid from another. These side chains determine whether amino acids are polar, nonpolar, acidic, or basic. These chemical properties ultimately influence how a protein folds and how it performs its function. Amino acids are connected to one another through covalent bonds called peptide bonds. A peptide bond forms through a condensation reaction between a carboxyl group and an amino group. Repeated peptide bonds create the characteristic N-C-alpha-C backbone of a polypeptide. Every polypeptide has direction, running from its N-terminus toward its C-terminus. Peptide bonds are rigid and planar, limiting rotation and influencing the shapes a protein can adopt. The primary sequence is simply the order of amino acids within the polypeptide chain. The chemical properties of the side chains provide the information that drives the protein toward folding. Folding produces a unique three-dimensional structure that allows the protein to function. As Anfinsen demonstrated, the primary sequence contains the information required for native folding under physiological conditions. Protein structure is organized into 4 hierarchical levels: primary, secondary, tertiary, and quaternary. Primary structure is the exact linear sequence of amino acids in a polypeptide, written from N-terminus to C-terminus. This sequence is genetically encoded and provides the foundation for every higher level of protein structure. Even a single amino acid substitution can dramatically change protein function and contribute to disease. Secondary structure describes local folding patterns within the polypeptide backbone. An alpha helix is a right-handed coil stabilized by hydrogen bonds between backbone atoms. In an alpha helix, hydrogen bonds occur between backbone atoms every 4 residues, while side chains project outward. Beta sheets consist of extended strands aligned side by side and stabilized by hydrogen bonds. Beta sheets can form with strands running in parallel or in opposite, anti-parallel directions. Loops and turns connect helices and sheets and are often located on protein surfaces where they can participate in binding. Tertiary structure describes the complete three-dimensional arrangement of a single polypeptide chain. Tertiary structure is stabilized by interactions between side chains, including hydrogen bonds and ionic interactions. Hydrophobic residues tend to pack inside the protein, forming a compact core away from the surrounding solvent. Disulfide bridges are covalent cis-to-cis bonds that can help stabilize the three-dimensional protein structure. Van der Waals forces also contribute to the many interactions that stabilize the folded tertiary structure.
Quaternary structure describes how multiple polypeptide subunits assemble into one functional protein complex.
Hemoglobin is a classic example, consisting of 2 alpha and 2 beta subunits that cooperatively bind oxygen. DNA polymerase complexes contain multiple subunits that work together to support high-fidelity DNA replication. Antibodies are another example, with 2 heavy and 2 light chains forming the characteristic Y-shaped immunoglobulin. Proteins often contain recurring structural motifs, such as beta-alpha-beta loops and Greek key arrangements. Protein domains can fold independently, while folding generally buries hydrophobic residues inside and leaves polar residues facing the solvent. Thanks for watching the BioInsights. Subscribe and keep exploring the fascinating world of biology with us.